Hi, everyone. My name is Angela McDonnell, and I'm really excited to be a part of this rare plant genetics workshop. And today I'll be presenting an overview of molecular techniques for plant evolution and conservation studies. This PowerPoint and presentation were developed in collaboration with my supervisor, Nor Wickett. We both are working at the Chicago Botanic Garden. And we are on Team Shedia, working on Shedia genome assembly and evolutionary studies. And I have some pictures of Shedia here on the left. Shedia and people also affiliated with the project, excuse me. So our goals with this presentation are to provide you with an overview of the molecular methods and some of the data types that can be applied to systematic and conservation studies in plants. We're not going to provide you with a how, when, or why you might apply these methods. And our main reason for that is because that's one focus of the meeting, and we hope it will come up repeatedly in our discussions. And so we're more going to focus just on the techniques and some terminology and a brief overview of the methods. So one of the first things I wanted to touch on are that molecular data are really useful. And this is an idea that has been in existence for a very long time. It's one of the earliest studies was done by Church in the late 1800s, and he used molecular data to answer ecological questions using molecules. Specifically, he was looking at the terracin pigment in African turacos, which is a sort of bird, and it's only found in a family of birds with these special types of feathers. I guess I've read about it. I don't know anything about it personally other than this, but that's one of the earliest studies about molecular data, looking at how different birds are related. And he found it was only in this one family. And then we all probably know about Gregor Mendel around the same time, who was working with 7 characteristics of peas and crossing them to demonstrate that there were actions of invisible factors that affected how traits were passed down from generation to generation. And so they weren't looking at molecules necessarily, but they were beginning to develop that understanding that molecular characteristics are useful, even if they weren't using those terms. In plant conservation, molecular data have early roots in the 1920s. Vavilov, a Russian geneticist and plant breeder, proposed that crop improvement could proceed by drawing from the wide genetic variability that occurred in the wild relatives of crops. And so he sort of determined where there were concentrations of different varieties and used plant morphology, but also cytology and genetics to look at resistance to pests and other diseases, and also their climatic adaptations. And to transition into some of the methods, I also want to mention here that there were a lot of studies in the 70s and the early 80s by Hamrick et al. And others who were looking at the relationships between different plant traits and plant molecules that are sort of an extension of the genetic traits or isozymes. And so they were looking at how different enzymes in different plants could inform relationships and other aspects. And so some of these foundational techniques, the first one I want to talk about are isozymes and allozymes. And like I mentioned, these were introduced pretty early on in the 1970s. And allozymes refer to multiple forms of an enzyme encoded by different alleles, or if we're looking at one locus. And isozymes are multiple forms of an enzyme that are encoded by different genes. So in that situation, we'd be looking at multiple loci. And basically this method means that it proceeds by extracting protein from plant matter, and then those proteins are electrophoretically separated on starch gels. Those starch gels are then stained and scored, and you can see one of those to the right on the top. And the input is usually multiple fresh tissues are ground up together. It can be quite a bit of tissue. And the pros of this are that you're able to estimate genetic variation in natural populations using these enzymes. You don't need any a priori information about species or their genetic sequences, and you can relatively easily obtain information from multiple loci. The cons to this method are that the sampling is fairly destructive. You need a decent quantity of fresh plant material from all of your individuals. You often have low levels of polymorphism associated with this method because you're looking at the protein level and not the DNA level. And you're not going to get necessarily too many loci at one time. So from there, I want to move on to talking about other methods that use enzymes in different ways. So RFLP, AFLP, and rapids. Some of these use enzymes, not all of them. And these are all kind of coming onto the scene in the 1990s. And basically what they all have in common is that they're comparing DNA fragment patterns. So for RFLP, that means we're looking at restriction fragment length polymorphisms. AFLPs are amplified fragment length polymorphisms. So that's a method that uses PCR. And then rapids also use PCR, and that's a random amplified polymorphic DNA approach. So the input for this method would be total genomic DNA that's extracted from plants, from leaves, for example. And the pros to these methods are that you can estimate genetic variation in natural populations again. It doesn't require any a priori information about genetics or genes or anything. And you do get information from multiple loci at one time. Some of the cons, again, are that it can require a lot of DNA, a lot relative to some of the other methods I'll talk about later. You do often obtain lower levels of polymorphism. The bands that you get on these gels that you're reading, or the fragments that you are able to recover, are of unknown sequence. You're not able to attribute them to any genes or anything. And in part because these methods are reliant on restriction enzymes, or enzymes that chomp DNA, and PCR, different conditions, different enzymatic conditions or different reaction conditions can yield different results, and that can make studies difficult to compare. I want to talk now a little bit about microsatellites, another foundational technique that is a little bit more recent. So this is more common in studies from the 1990s. And this is where we're looking at repetitive DNA sequences. So short tracks of tandemly repeated or adjacent DNA motifs. And these are also known as SSRs, STRs, mini satellites, VNTRs. They have a lot of different names that you may or may not be familiar with, but they're all basically looking at short repeated bits of varying length. And the input to this is that total genomic DNA, again, the pros are that you would estimate genetic variation. They can be really useful for that. And the sequences are known from each sample. And so that is a big change from some of the earlier methods. And these tandemly repeated pieces of DNA are often highly variable, and they can be really variable between populations and also within populations to help you identify individuals. So really useful for genotyping. This method does use, is PCR based or polymerase chain reaction based, and Sanger sequencing. And PCR and Sanger sequencing are relatively simple techniques. They can be easy depending on how comfortable you are with the methods and your familiarity with troubleshooting those methods, but they can also be hard. So I had it as both a pro and a con. This method also does require some a priori information about species so that you can find these satellites or these repeated pieces of DNA across all of your samples and amplify and use the same orthologous loci across all of your samples. So now I mentioned PCR and Sanger sequencing are both easy and hard. And now I want to kind of step back a little bit and talk about polymerase chain reaction. So this is a technique that was developed in 1983, and it's been widely applied to a lot of different studies and situations. But basically what this means is that you extract DNA from tissue, and then you use primers around a piece of a locus of interest. So you design some primers, and those are shown in this figure in purple, if you can see, or to the lower right of the DNA template sequence of interest. You design short primers that stick to either side or end of that template region of interest. And then you add heat to separate your double-stranded DNA into 2 single strands. You lower the temperature to allow those primers to anneal to this single-stranded DNA. And then a DNA polymerase enzyme will help to extend that complementary sequence. And the cycle is repeated many times. So you're basically making like many, many, many Xerox copies. You can make millions to billions of copies of your original piece of DNA in a relatively short span of time, in an hour or so. And so this has really broad applications. You put in, your total DNA, total genomic DNA in a tiny amount. And so that's a pro. You can use, 0.1 microliter of your very dilute DNA. And like I said, this has broad applications. PCR can be really easy and quick once you know what your cycle conditions should look like and what your recipe for combination of DNA primers, DNA template, and your enzyme are. But it can also be really hard if you don't know exactly what those are. It is a locus by locus approach. So when you're doing these reactions, you need to isolate the one locus for one sample separately. And so you do many PCR reactions to complete a study typically. And it does require some a priori information about species, since you need to know what those primer sequences should be. PCR is often followed by Sanger sequencing. Sanger sequencing was developed a little bit earlier than PCR in the 70s. And so to complete a Sanger sequencing run, you would have DNA, you would amplify that DNA through PCR to create lots of copies of what you want to sequence. You could also clone the DNA, but instead of doing PCR, you would then do a PCR-like reaction to prepare your copies of DNA for sequencing, and that's basically adding fluorescent terminator nucleotides to the end of your molecules to generate molecules of different sizes that have those terminator nucleotides that are fluorescent. And then you basically read the identity of each nucleotide in order from the last nucleotide of the shortest fragment to the longest. And that's shown in 1 and 2 of the figure here. And your input to this reaction is a pretty small volume of DNA from a PCR reaction or from a cloning experiment. And the pros here again are that you know what all these sequences are, and you have a lot of control over the quality of your data in that respect. And the data are really widely useful, especially for phylogenetics. So if you want to estimate relationships among species, people have done this for plastid data or nuclear data for various loci. And Sanger sequencing can be really easy and quick. You can complete PCR and Sanger sequencing, in one day if you'd like to. But the cons here again are that it is a locus by locus approach, and not all of the loci that you're going to be using for this are variable. And Sanger sequencing, as it is a PCR-based approach, can be really difficult to troubleshoot. So there are some drawbacks. So why would we possibly need some newer techniques, or why are the newer techniques that I'll talk about next useful? Well, they yield a lot more data per unit of time spent, and often per unit of money too. And they give you more data. More data better represent the entire genome of the organisms that we're studying and give us a better idea of the evolutionary history, and can help us avoid bias or errors that are caused by using only one locus. It's also cheaper per unit. As of August of 2020, 1 million bases of raw data cost us roughly 0.8 cents. It's also flexible. So these methods often allow us to use degraded samples, DNA extracted from herbarium specimens or historical specimens, for example. They still have pretty fast sample processing and turnaround, and that's in large part because we can multiplex, or we can process samples in batches and sequence them together and obtain data for hundreds to thousands of regions for many samples simultaneously. And this has been enabled by high throughput or massively parallel sequencing. So next generation sequencing is likely something that you've already heard of, and that's as opposed to Sanger sequencing. So this refers to a number of platforms and companies that allow the sequencing of millions of DNA fragments in parallel. And in general, the sequences that are generated from these methods are relatively short, so between 100 and 800 base pairs. And these include methods that are like the Roche 454, Ion Torrent, and Illumina sequencing. Illumina sequencing is by far the most common. And these methods, interestingly, generate roughly 10,000 times more data per day per machine versus Sanger sequencing. And I'm showing that here with that red arrow on that graph. So the methods for next generation sequencing works in a very similar way to Sanger sequencing. So you have to prepare your DNA. So you'll extract it from dried tissue. You'll fragment it to the desired size, that's different from Sanger sequencing. You'll fragment it to the desired size through either sonication or enzymes. And then you're going to add adapters and barcodes to the ends of these fragments. And that's shown in the second. So we see genomic DNA, and then there's some things that happen: bead purification and quantification and repair, blah, blah, blah. We don't need to talk about that. But below that, where we're adding these adapters and barcodes, those are going to help us identify the samples and track them at the end of the experiment. And the collection of the fragments at the end of this experiment is a sequencing library. The library preparation method will be different depending on the type of sequencing that you do. And so here we're showing what it might look like for an Illumina sequencing run. When you want to sequence your library, you will sequence the inserts, the piece of DNA that you inserted between those two adapters. And each insert is a unique sequence. The library that we generated is attached to a flow cell, and that is the green thing, the slide looking thing in the upper right, or the upper middle right of this figure. In each adapter, each molecule, insert plus adapter, each molecule is amplified on this flow cell. The sequence of each amplified insert is then read by the sequencer. The sequence of that insert is a read. So we have inserts that are from these libraries, and then the data that comes from the insert is a read. For Illumina sequencing, we may get hundreds of thousands to hundreds of millions of reads. So those are our raw data. In a perfect world, we would want to sequence and assemble complete genomes for everything and just be done with it. And we can use next generation sequencing to help us achieve that goal. However, there's a huge amount of variability in the genome size of plants. And I'm showing that with a red bracket at the top. So especially for angiosperms, we can see that the amount of DNA in cells, or the corresponding genome size, is hugely variable. So complete genomes for all individuals in a study is not yet economical, efficient, or feasible. To access the data set that samples broadly from the genome, I'm going to talk next about reduced representation methods. So these methods reliably sequence a smaller fraction of the genome in a way that we can collect the same fraction from each species or individual that you include in a study. One method is transcriptomics, and I'll talk about that just briefly here. So for example, by sequencing the messenger RNA, you can target only the expressed fraction of the genome and sequence a largely overlapping set of the same genes for different species or individuals. But extracting and working with RNA can be difficult, and you may not need all of the approximately 30,000 expressed genes that result from a transcriptomic study. The next method of reduced representation sequencing I want to talk about is low coverage genome sequencing, or you may have heard of genome skimming. And this was developed around 2010, and that the libraries that we generate are reduced so that only the highly repetitive genome regions, such as ribosomal DNA and organelle genomes, are recovered. And this is often known as shallow pass genome sequencing. And I'm showing on the figure on the right with the arrow, if we consider the top of the pyramid shallow sequencing, and the bottom of the pyramid is the deepest possible sequencing of the genome that we can do. If we sequence more shallowly, we can detect things like ribosomal DNA, the plastome, rearrangements and repeats, mitochondrial genes, and those sorts of things, but we won't necessarily get at the conserved nuclear loci or the entire genome. So for this method, you need up to a microgram of extracted DNA, so not too much. You can get that from, half a gram or less of a leaf, dry weight. The pros is that this method is relatively inexpensive. It can be used in combination with hyb-seq, which I'll talk about next. The data from this method can be combined with other types of data. So if you do a couple of different genome scans for the same individual, you can combine those data. The cons are that it's not always useful for answering species level questions because you're not necessarily getting super variable regions. So we could talk about the differences between plastid and nuclear data later, if that's something that people are interested in. You don't always obtain hundreds of loci because you're only getting a few loci if you consider a whole plastome one locus and a ribosomal DNA cistron one locus. You might only consider that you're getting reliably a handful of loci. And you're not actually using a lot of the sequence that you obtain. So not all of your sequence that you get from this method is considered useful. So there's some waste there. I mentioned on the last slide hyb-seq. So hyb-seq was developed around 2010 and has become a standard in many plant molecular studies at present. And here, genomic DNA libraries are reduced to DNA regions of interest, such as highly conserved regions, low copy or single copy nuclear loci, or even functional genes. It doesn't require too much DNA. And this method, basically, you take your sequencing libraries, which are shown at the top in navy blue and orange, you denature those to be single-stranded, you bind your single-stranded library portions to complementary baits or probes that target these conserved regions. And these are biotinylated, and the biotin binds very readily to streptavidin. So these baits that have biotin on them can be stuck to a magnetic bead, and these bead bait and DNA hybrids can be separated from the other fragments that don't target the regions of interest. And so the things that you don't want to sequence are washed away, and the things that you do want to sequence are retained. And so the beta DNA is sequenced and often includes the target regions and the adjacent non-target DNA regions. So this method allows us to generate lots of data for many samples at once for hundreds or thousands of long independent loci, including more variable non-coding regions. And that's the non-target DNA that I mentioned a little bit earlier. This method is often used in combination with genome skimming, and the data from this method can be combined with data from other studies for the same loci. So this is a highly repeatable method. One of the cons of this method is that it is pretty expensive up front. So some of the reagents and the tools that you need to do this in your own lab can be pretty pricey. However, the price per sample of this method is not too terribly high. It can be between $22 to $70 per sample. It does require resources to design the baits, though there are some universal bait kits that exist and are cheaper to use off the shelf. Rad Seek was developed between 2006 to 2012, there are a number of papers on the method. And this method reduces the libraries to regions adjacent to restriction enzyme cut sites. And the number of resulting fragments that you get and that you sequence depends on both the enzyme that you use as well as the genome that you're putting in. There are a few different flavors of this, so it might just be called RAD or restriction associated digest sequencing. Double digest RAD, which uses additional a two enzyme method, or genotyping by sequencing. So in this method, you input between 500 nanograms and 2 micrograms of extracted DNA from a leaf, for example. You use restriction enzymes to chop it up, that's shown at the top. The cut DNA that you generate is made into a sequencing library. You can pool those samples and sequence them together to get your raw data. And when you assemble your data after rad sequencing, you get little stacks of short loci that contain SNPs or single nucleotide polymorphisms. So you're able to generate homologous regions for many samples at once. It's fairly economical and it's very scalable for population level analyses. It's a really great way to get lots of SNPs. Some of the cons are that the loci tend to be pretty short and can have only one or a few SNPs or single nucleotide polymorphisms per locus. Missing data in this method can be problematic. It's difficult to generate the same set of loci for independent runs. There can be a batch effect because you're using enzymatic methods that you would want to process all of your samples, all of your DNA samples with the same enzyme at one time and not necessarily combine different runs. It can be difficult to assess orthology or which loci go together across samples without a reference genome because the loci that result are short. And this method should really only be applied to populations or close relatives, which is a con, but it could also be a pro since, I'll leave it there. It could be a con, but it could also be a pro, depends on your question. So you're going to be hearing a lot more about many of these techniques and probably some others throughout this workshop. We've compiled some references in a file that is available to you for additional information about each of these methods. And I'm happy to gather more papers if that's something that people are interested in. We'll also be participating in this workshop. And so as any questions arise, I'm happy to answer them about any of these methods. And we're also going to develop a sort of crib sheet throughout this workshop in a Google Drive spreadsheet that is responsive to the questions and discussions that happens throughout the workshop. And I'll provide a link to that later during the workshop. And we really hope that it will provide attendees with a resource that can be referenced as needed, when needed, whenever, and as you all are doing your work. So with that, I want to say thanks for your time and attention, and I'd be happy to take questions now, later, or whenever. Thank you.